Hi, everybody. My name is Haley Sutton, and I'm going to be talking about the project that I did. And that project is the characterization of a bacterium isolated from acid mine drainage. So, in Pennsylvania alone, there are over 11,000 abandoned coal mines that affect Pennsylvania water. In this experiment, an isolate of bacterium was obtained from a local park, Boyce Park to be exact, which isn't actually that far from California University of Pennsylvania, just to see what is exactly in our water and if we should be alarmed with what is actually in there. So the unknown bacterium was analyzed using KBase. Now, KBase is used to sequence unknown bacterium that you find in the field against species of bacteria with known characteristics. Now, when you compare the two bacteria, like the unknown to the known, you can see hopefully a close relative, or maybe even narrow down your search on what you actually found to a specific bacteria in the best case scenario. The isolate was tested for antibiotic as well as metal resistance, and metabolic properties were looked at in order to narrow down what treatment would possibly be needed for this specific bacterium if treated in the field. Now, the more research that is done on these acid mine drainages would hopefully broaden the horizon on what researchers know in order to combat the unknown of biological impacts. So if you look at figure 1, it shows how our plates looked before we ran an experiment. So each student was assigned a different unknown isolate to be analyzed. In this particular experiment, my isolate was B6. So in order to perform the experiment that I designed, a few things were needed. I needed R2A media plates, which I needed 6 in total. I needed 2 for control, 2 for one type of metal, and 2 for a different type of metal. So I had 6 sterile discs on each plate, so there was 36 total sterile discs. Now I needed the zinc sulfate as one metal that was going to be compared. I needed cuprous sulfate as the other metal that I was going to be compared, pipettes, and a control strain. So if you look at figure 2, that's a positive control. So this positive control was going to be used as reference for growth. There was also a negative control that should have showed no signs of growth. Those are just to make sure you had something to compare to that you would have known results. So the 6 R2A plates were prepared. There were different concentrations of zinc on 2 plates, different concentrations of copper on 2 plates, and as well as 2 control plates. Now, our results were measured looking at the zone of clearing, and that zone of clearing is where things will choose not to grow because of the presence of those metals. Now, these plates were incubated at 30 degrees Celsius for 48 hours, and the results are photographed in figure 3 and figure 4. Figure 3 shows the plates coated with zinc in different concentrations, and figure 4 shows the plates coated with copper in different concentrations. Now, in figure 3, if you look, then you will see that inhibition at highest concentration was minimal for zinc, but it was still there. In figure 4, inhibition at the highest concentration is there. It's more so for copper than zinc, but it's still minimal. The control strain was compared with B6 growth, and the controls underwent the same process to see the difference the metals would potentially make in those plates and inhibit possibly their growth. So let's talk about our results. At the highest concentration levels on both different plates with different concentrations of metals, there's inhibition, but that inhibition is minimal. And neither metal showing inhibition suggests that there is a high tolerance to metals within this isolate. So in conclusion, the B6 isolate showed little to no inhibition to metals, suggesting that this isolate was resistant to metals. Upon analyzing this isolate's genome, sequencing through KBase, the isolate's closest known relative was found to be Serratia, which is a facultative anaerobe and opportunistic pathogen. Now, I want to give a special thank you and shout out to my professor, Dr. V. She performed this experiment for me since we were not able to be in the lab due to COVID, unfortunately. And she performed it in a very smooth and efficient way and showed precise results. And I just couldn't thank her more for doing that for me, despite not being able to be in the lab, unfortunately. So if anyone has any questions, please feel free to email me and I'd be happy to try to answer them for you. I hope you enjoyed. Thank you.